Today I will be talking about cerebral oxygen metabolism during hypercapnia challenges. Cerebral metabolic rate of oxygen, or CMRO2, is the rate at which the brain consumes oxygen, and it's considered a sensitive indicator of brain viability and function, more than oxygenation level alone or cerebral blood flow alone. It can be calculated using the Fick principle, which states that the oxygen consumed by a tissue should be equal to the difference between arterial and venous oxygen concentration multiplied by the cerebral blood flow. Having the means to quantify CMRO2 would have an important part in understanding cerebral oxygen metabolism, both in health and disease. So the current gold standard to quantify CMRO2 is PET, but over the past years, many MRI and near-infrared-based methods have been developed as well. In some of these MRI methods, hypercapnia challenges are used as a calibration procedure. And to have a reliable quantification of CMRO2 in such studies, it's important that the applied hypercapnia does not affect the measured CMRO2. In normal physiology, we know neurovascular coupling is tightly controlled between blood flow and oxygen consumption in order to supply needed substrate and remove end products. It's known that induced hypercapnia increases cerebral blood flow. However, there have been conflicting reports about whether CMRO2 is affected or is maintained at normal levels during such an increase of CBF and oxygen delivery. It's been also suggested that oxygen consumption on the mitochondrial level might be involved in the regulation mechanism. The enzyme which is responsible for the majority of oxygen consumption in the cell is cytochrome C oxidase, which is the fourth complex in the electron transport chain in the mitochondria. It accepts and donates electrons and changes its redox state from oxidized and to reduced, depending on the electron's flow and oxygen availability. By quantifying the redox state of this enzyme, we can learn about the oxygenation level in the cell and the mitochondrial status. So in this study, we wanted to determine whether CMRO2 and the redox state of cytochrome oxidase would change in the mouse cortex when CBF increases due to induced hypercapnia. For this, we applied a new multimodal technique that combines near-infrared spectroscopy with MRI to simultaneously quantify cerebral blood flow, CMRO2, and redox state of cytochrome oxidase non-invasively from the cortex of mouse models. So a little bit of background about near-infrared spectroscopy. Near-infrared light can penetrate and travel deep in the biological tissue. When we shine light into the brain, the light is either absorbed by light absorbers or scattered in the tissue. From the detected light, we can extract useful information regarding the composition of the tissue and the concentration of important absorbers like hemoglobin and cytochrome C oxidase. Hemoglobin data, which includes deoxy, oxy, and total hemoglobin, can provide information about the oxygenation in the tissue, or StO2. When combining StO2 with arterial oxygen saturation, we can calculate the oxygen extraction fraction, or OEF. By combining these measurements with CBF values, we can quantify CMRO2 using the Fick principle. As I mentioned before, cytochrome C oxidase can be either in its oxidized or reduced state. These two forms can be distinguished with NIRS because they have different optical properties in the near-infrared window. We have recently published a paper that describes the concept and the algorithm used to quantify cytochrome oxidase for those of you who are interested in more details. To quantify cerebral blood flow, we applied arterial spin labeling MRI to generate perfusion map. CBF was calculated from a region of interest in the cortex, which is estimated to be similar to the region of sensitivity for NIRS. This is a schematic design of the NIRS-MRI system. Two optic fibers are positioned on the shaved head of the anesthetized mouse to shine and collect light from the cortex. During data acquisition, the mouse with the fibers is stabilized inside the MRI coil for imaging, and the two systems were operated independently. During data acquisition, the mice were anesthetized, spontaneously ventilated, and positioned in the MRI for imaging. NIRS data were collected continuously, and ASL imaging was applied first at normocapnia, and then during the inhalation of 5% CO2 to induce hypercapnia and to create perfusion maps at each condition. This is a representative example of the perfusion maps from the same animal acquired before and during the CO2 challenge. We can see here hyperintensity in the cortex during the inhalation of CO2. From these maps, we quantify the cerebral blood flow values from the region of interest. Each symbol here is another mouse. We found that CBF increased significantly during the inhalation of 5% CO2. These data are consistent with the reported relationship between CO2 levels in blood and the regulation of CBF. An increase in arterial CO2 tension causes vasodilation, which leads to the increase in the blood flow. We also saw a significant increase in the oxygen, in the tissue oxygen saturation. Oxygen extraction fraction decreased significantly while CMRO2 remained constant. This shows that during the hypercapnia challenge we applied, oxygen metabolism in the cortex did not change, and that the CBF-CMRO2 coupling was intact since OEF decreased to maintain stable CMRO2. In terms of the enzyme cytochrome C oxidase, there was a significant increase in the oxidized form and a significant decrease in the reduced form. This means that the total content of the enzyme did not change, but its redox state, which is calculated as the difference between the oxidized and the reduced form, increased during hypercapnia. In addition to the change in the baseline level, it's important to understand the relationship between the measured parameters. So we found a positive correlation between the redox state of cytochrome oxidase and STO2, a negative linear correlation between the oxygen extraction fraction and the redox state. However, there was no correlation between the redox state of the enzyme and the cerebral blood flow. These correlation graphs shows that under hypercapnia, the redox state of cytochrome oxidase was not affected directly by the cerebral blood flow, but it's affected by the availability of oxygen and the ability of the tissue to extract it from the microvasculature. To summarize, on one hand, hypercapnia caused vasodilation and increased cerebral perfusion. On the other hand, it led to additional oxidation of cytochrome C oxidase, but there was no linear correlation between blood flow and the redox state. This ended up with less oxygen extracted from the blood, which means lower OEF and higher STO2. And because of the reduced OEF, CMRO2 remained constant during this hypercapnia challenge. It seems from these data that some kind of regulatory adjustment takes place at the mitochondrial level. For example, adjustment of cytochrome oxidase affinity to oxygen that holds the rate of oxygen consumption constant despite the increase in cerebral blood flow. To conclude, we showed that CMRO2 remained constant under a mild hypercapnia challenge. Also in this study, we applied the multimodal NIRS-MRI system, which enabled non-invasive quantification of CMRO2 in the cerebral cortex of mouse model in vivo. In addition, this technique allowed the quantification of multiple physiological variables simultaneously, such as CBF, cytochrome oxidase, STO2, and OEF. And having such a multimodality system will open new possibilities for studying oxidative metabolism and mitochondrial damage using many mouse models of brain disease available. I would like to acknowledge Dr. Jeff Dunn's lab and the Experimental Imaging Center at University of Calgary and our funders. And thank you all for listening.